Top 10 Present 10 Amazing Ways We Study the Diets of Our Ancestors. When an archaeological site is discovered, the question "what food did these people eat?" might sound unexciting, especially to those used to the idea that archaeology is about discovering lost temples filled with hidden doors and haunted treasures. But understanding what our ancestors ate means understanding their subsistence strategies and their relationship with the environment. The cumulative effect sheds light on some big questions of the human past, such the transmission of technological innovations, cultural contact, and even the spread of agriculture. Number 10. Teeth marks. Teeth, made of resistant tissue, tend to survive remarkably well. The food we eat leaves microscopic marks on tooth enamel, and the length and orientation of these marks depend on the food we consume. Modern Inuit groups in Greenland, whose diet is largely based on meat, display mostly vertical marks on the lateral surfaces, while groups living on an almost exclusive vegetarian diet display shorter marks, both vertical and horizontal. Number 9. Remains of individual meals. Under some rare and incredibly fortunate circumstances, archaeologists can find meals, almost intact. The ancient city of Pompeii is arguably the most famous example of this kind. Many meals were found virtually untouched, still served at the table and preserved for centuries under thick layers of volcanic ash. We have also identified entire food shops with their products still suitable for recording. Number 8. Animal remains. Animal traces such as bones, antlers, and shells provide useful information on past diets. Based on animal bone samples, specialists perform statistical analysis on the age, sex, and the season of the death of the animals. Number 7. Digestive tract contents. The stomach is made of soft tissue and survives only under rare circumstances, such as extremely dry climate or cold temperatures. Sometimes, it is possible to retrieve samples of food traces from the stomach and other areas of the digestive tract, including the colon and even the intestines. Number 6. Tooth decay. Changes in our diet has affected tooth decay rate significantly. Refined sugars and starchy food has elevated the overall incidence of tooth decay and encouraged a specific pattern of tooth disease and tooth loss. Starch makes our teeth more vulnerable to dental disease, so societies that rely on cereal consumption diet present a higher rate of tooth decay. Number 5. Fecal material. Some organic traces survive after they have passed through our alimentary tract. The brave specialist who digs into the secrets held by ancient poo is sometimes referred to as an analyst of desiccated paleofecal matter. Number 4. Food processing tools and equipment. Food procurement and processing is normally assisted with tools. Evidence of fishing, for example, comes in the form of hooks, fish spears, fish traps, and nets. Stone tools used for butchering animals display a specific microscopic wear pattern, and this evidence can be cross-referenced with animal bones assemblages found at the site. Number 3. Isotopic methods. We are what we eat. At least this holds true for isotopic methods, which rely on chemical traces that the food we eat leaves in our bodies. One of these techniques reads the ratio of nitrogen isotopes found in bone collage. Nitrogen-15 increases as it travels up the food chain, while nitrogen-14 decreases. Individuals who depend largely on cereals and vegetables display a low nitrogen-15 to nitrogen-14 ratio compared to individuals who eat animal meat, blood, and milk. Individuals who rely heavily on a marine diet display even higher ratios of nitrogen-15 to nitrogen-14, since the marine food chain has a higher number of levels compared to terrestrial environments. Number 2. Botanical remains. Phytoliths are minute botanical particles specific to certain plant species and may come from the flower, stem, or root. These particles can be recovered from sediments, pottery fragments, the surface of human teeth, or attached to stone tool edges. Phytoliths can identify specific botanical species linked to food consumption and differentiate between wild and domestic plant species. Number 1. Tartar. Tartar can be a truly informative source of past diets. As it builds up on our teeth, traces of food get trapped in it. Dentists who scrape tartar are a recent privilege, so our ancestors had a lot of tartar on their teeth. Tartar is especially informative on the plant species consumed, since pollen grains survive a long time. Microscopic fragments of fossilized plant species and bone fragment particles can also be found buried in the tartar.